Okay. So the topic of the today lecture is fluorescence and confocal microscopy. So you can see what is microscope. It is an instrument to see a small object with the naked eye. Why we use a microscope? Because from our naked eye we cannot see a small objects like atoms, molecules. So we need a microscope. You can see different type of light microscope, compound, fluorescence, and confocal microscope. There are few terms. So there is a microscopic scale where we can see from our human eye, we can see the size object up to 100 micrometer. We can use the light microscopy to see the size object up to 1 micrometer, and for electron microscopy we can see the object up to 1 nanometer.
आप इधर से बताइए these are the mouse kidney cells they are staying with.
In the picture, this is a picture of mouse kidney cells. The cells which are stained with the different dyes. One is Dapi, that is fall in the blue region. Second is Calodin, that is fall in the green region. And third is Rhodamin B, that fall in the red region. So you can see the images, how clearly and how sharp this we can see clearly these images. So this is the kidney cell. Next, you can see also the HeLa cells. You can see the dark images of HeLa cells, and then there is a staying dark cells.
Yes, sir.
I am Nihit Saigal. I am an Rice Fellow at the European Molecular Biology Lab at Heidelberg in Germany. And today I am going to discuss the basics of bioimaging techniques. So I would like to start with one quotation from a famous physicist, Richard Feynman, who wrote an article about saying that there is plenty of room at the bottom. What Feynman was referring to was that we have enough space at the scale which is not visible to our naked eyes. At the nanometer or molecular scale, there is enough room to manipulate and engineer things over there. But the first step in doing this is to be able to see things at that scale, and that is where the microscopy becomes very important. So this is the outline of my presentation. First of all, I will discuss the basics of bioimaging, then I will introduce fluorescence microscopy. I will show you some practical considerations in fluorescence microscopy. Then I will discuss the advanced fluorescence microscopy techniques, and finally give you an introduction to EMBL and my current work. So this is the first part. We learn about the atomic model, where we know that the atoms consist of a nucleus with electrons going around them in the form of orbitals. What happens when you shine light on an atom? What happens is that the electron jumps from one orbital to another orbital, let's say from the ground state to an excited state, but the excited state of the atom is unstable, which means that the electron wants to jump back to the ground state, and in this process it emits light with some energy, and this process is essentially called fluorescence. It is also possible to understand this process in the form of this energy diagram, where the different states of the atom, of the electron, are described by these different levels. And the process of exciting the atom is shown as the electron going from the ground state to an excited state. And as you can see that it relaxes, the electron first relaxes via some vibrational excitations, and then it goes from the excited state back to the ground state, and in this process it emits energy in the form of fluorescence. So this is the basic process of fluorescence, and one thing that you can clearly notice is that the energy which is used to excite the sample is that the electron is much higher than the energy at which the emission happens, and that's why if you look at the intensity versus wavelength plot, you would see that the excitation spectrum is at a lower wavelength or higher energy, while the emission spectrum is at a higher wavelength or